All right, let's dive right in. Today's explainer is basically a high-stakes biological detective story. You see, for the past couple of decades, geneticists have mapped out thousands of genetic coordinates that are associated with complex human diseases. So we have this massive lineup of genetic suspects. But here's the problem. For a huge chunk of these suspects, we completely lack the motive. We know they're at the scene of the crime, but we don't know exactly how they're causing the disease. So today, using an incredibly detailed large-scale evaluation from the journal PLOS Genetics, we're going to look at the tools researchers are using to actually crack this case. And we are going to demystify what scientists call the colocalisation gap. Okay, so the crucial point is right here, 90%. Over 90% of the genetic variants associated with complex traits and diseases, these are variants discovered through massive genome-wide association studies, or GWAs, are found in the non-coding regions of the human genome. They don't just sit neatly inside a gene that tells us all how to make a protein. Instead, they're hiding out in the dark matter of the genome. Because of this, they're essentially just genetic coordinates with absolutely no obvious function. We know a coordinate is linked to a disease, like multiple sclerosis or Crohn's disease, for example, but we have no clue which specific gene it's actually messing with. And this matters immensely, because connecting that coordinate to the right gene, well, that's literally the very first step in developing a cure for those illnesses. So to figure out the how and why, scientists use our primary detective tool, colocalization. I want you to think of this kind of like a real estate investigation. We've got two pieces of information. First, we have our GWAS data, which tells us a genetic variant is linked to a disease trait. Second, we have eQTL data. Now, an eQTL is really just a genetic variant that controls the expression level, think of it like a volume dial, of a nearby gene. Colocalization is a super rigorous statistical method that tests whether the disease variant and the eQTL variant live at the exact same molecular address. If they share the exact same causal genetic variant, we can confidently say, aha, this genetic variant causes the disease by changing the volume of this specific gene. It basically nominates our target.
Section 1. Mind the colocalization gap. But, like any good detective story, we've got a major complication here. Even with our absolute best statistical tools and massive data sets, our investigations are just coming up short. Looking at the sheer scale of the mystery, researchers systematically analyzed over 1.3 million colocalization tests from the Open Targets Genetics database. That's the largest available public data set of its kind. And the crazy thing is, only 50% of the disease-linked GWAS peaks actually colocalize with a gene. That leaves us with a 50% totally unexplained gap. And it gets worse. When the researchers zoomed in specifically on immune-mediated diseases, things like type 1 diabetes or inflammatory bowel disease, only 34% of the genetic signals could be linked to a target gene. I mean, that is a massive colocalization gap. So why the gap? Well, the researchers started hunting for patterns, and they actually found a few key clues. First up, colocalization is far more likely when the genetic variance is physically closer to the gene it regulates. Makes sense, right? Second, more common variants, the ones with a higher minor allele frequency in the population, are just much easier to link, simply because we have more statistical power to actually see their effects. And third, context is absolutely everything. You get vastly better results when you test within the specific tissue relevant to the disease. Like, testing a cardiovascular genetic variant in a blood vessel cell is obviously going to work much better than testing it in a skin cell.
Section 2. The sample size trade-off. You see, the way a study is actually designed completely changes our ability to solve this mystery, and it forces researchers to choose between two less than ideal extremes. On one hand, we have high power, low granularity. Think of massive meta-analyses, like the eQTL Gen study, which looks at whole blood samples from over 30,000 individuals. This raw statistical power is incredible for finding rarer genetic variants. But whole blood is basically a soup of dozens of different cell types, which completely blurs the biological context. On the other hand, we have high precision, high granularity. These are your sorted or single-cell studies. They give us beautiful, precise biological context, telling us exactly what a gene is doing in a very specific type of immune cell. But because these studies are incredibly complex and expensive, their sample sizes are, well, they're tiny, often under 1,000 people. So they simply lack the statistical power to detect those rarer signals.
So why does that high precision matter so much? Well, the data here is wild. When you look at the enrichment for colocalization across different immune cells, certain signals jump right out. For instance, Crohn's disease and inflammatory bowel disease signals peak specifically in monocytes. Or look at lupus signals. They peak in B cells and dendritic cells. The data actually reveals that over 55% of colocalizing genes are detected in only one specific nominal cell type. If you're just looking at that whole blood soup, you're going to completely miss these subtle cell-specific signals. You absolutely need the precision, but right now, buying precision means sacrificing power. Section 3. One peak, multiple genes. Okay, wait, here is the plot twist. What happens when our statistical tools point to not just one gene, but multiple genes at the exact same time? The researchers found that roughly 47% of the peaks in the open targets data and 37% in the immune-mediated disease data colocalize with 2 or more different genes, almost half. So we really have to ask, are these just statistical false positives, or are we looking at some really complex shared biology here? To figure this out, the team ran some incredibly rigorous sensitivity analyses. First, they totally ruled out mapability errors, meaning this wasn't just a sequencing glitch where the computer got confused by 2 similar-looking genes. They also proved that random false positives just couldn't statistically explain the sheer volume of these multi-gene hits. Instead, they found actual biological evidence. When a variant links to multiple genes, the expression of those genes is highly correlated. And using some really advanced predictive models, like the SCGG model, they found these genes often share the exact same enhancer-promoter interactions. It's actual biological co-regulation, which is pretty amazing.
Let me explain exactly what that looks like in the data. When we look at these multi-colocalized genes, we see their expression correlation is significantly higher than your average control genes. They literally turn up and down together. Furthermore, these multi-colocalized genes are physically much closer to each other on the DNA strand than control pairs. A really great example from the study is a multiple sclerosis genetic signal that colocalized with both the CD5 and CD6 genes in CD8 T cells. Those genes sit right next to each other, they are highly co-expressed, and an enhancer loops over and controls both of their promoters simultaneously. So our single genetic suspect is actually hitting 2 targets at once. How cool is that?
Section 4. Closing the genetics gap. So this co-regulation idea entirely changes how we interpret GWAS data. Finding the causal gene might sometimes actually mean finding the causal genes, plural. So how do we adapt to this? Based on this massive evaluation, the researchers laid out a really clear roadmap for the future of genetics. Step 1. We have got to stop relying solely on easily accessible, healthy tissue. We must target diverse, disease-relevant cells, and crucially, we need to sequence cells from actual patients in disease states. Step 2, we have to maximize granularity. As we just saw, bulk tissue hides the answers. We need that single-cell precision to catch those sneaky, cell-specific regulatory tricks. And finally, step 3, we need to build massive sample sizes for these single-cell studies. We essentially have to pair the immense brute force scale of global meta-analyses with the granular precision of single-cell biology. It's an enormous logistical and financial challenge, for sure, but it is literally the only way forward. Which kind of leaves us with this final, really provocative thought. Will combining massive scale with single-cell precision finally close the colocalization gap for good? Doing this isn't just some academic exercise. Filling this gap is exactly how we translate mysterious genetic coordinates into the life-saving, targeted drugs of tomorrow. It is quite literally the future of genomic medicine. So what do you think about this single-cell revolution? Let us know. And hey, if you enjoyed this explainer and want to keep decoding the cutting edge of science with us, make sure to like and subscribe to @Beyond Biology Lab. I'll see you in the next explainer.